Hi everyone, this presentation covers Chapter 16 on Research Protocols in Jacobsen's third edition of the textbook, Introduction to Health Research Methods, a Practical Guide. In this chapter, you will learn that a research protocol is a detailed guide describing all actions that will be taken during the implementation of a research project. Here's an outline of the chapter. Section 16.1 provides an overview of research plans by study approach. Section 16.2 covers writing a research protocol. Section 16.3 covers rigor and reproducibility. Section 16.4 covers research timelines. Section 16.5 covers roles and responsibilities. And Section 16.6 covers preparing for data collection. So here's an overview of research plans. A research protocol is a detailed written description of all of the processes and procedures used in a research study. This includes information for recruitment of subjects, data collection from subjects, and the data analysis. For all types of studies, including the primary, secondary, and tertiary study designs, it is helpful to create a research protocol that will guide each step of the data collection, management, and analysis process. Figure 16.1 covers research plans for primary, secondary, and tertiary data collection. Here you have different tasks that are required for primary, secondary, and tertiary studies. It goes over defining the study question, then selecting approaches and data sources, ethical review if needed, data collection, analysis, and reporting. You can go over the details of each of these data collection types on your own. Section 16.2 covers writing a research protocol. A comprehensive research protocol describes the exact procedures that will be used for every step of the research process. These protocols should anticipate foreseeable challenges or areas of potential confusion and address them as completely as possible. A strong protocol provides enough detail that another researcher could easily replicate the study in a new population. Figure 16.2 goes over some examples of items for primary study protocols. So, for example, the goal that is included in a primary study protocol is the main goal and specific aims of the study. You also want to include the study design, which has details on the key features of the study design and key variables. So it is helpful to include whether the study is, say, a cross-sectional, a cohort, case control, or experimental, written explicitly in the protocol. Then it also goes over things like sampling, sample size, recruiting, consent, the questionnaire, additional assessments, data management plans, confidentiality, and data analysis procedures. Section 16.3 covers rigor and reproducibility. Here we have the definitions of both of these concepts. So the rigor for the research study really is the careful design, implementation, interpretation, and reporting of the actual results, and in alignment with ethical research protocols. Reproducibility is the ability of an independent researcher to implement another researcher's data analysis protocol and generate the same results, but using either a different population, or if it's a secondary study, actually just using the original data set and being able to replicate what has been done. And this really includes a detailed set of explanations in the protocol. Section 16.4 covers research timelines. Research timelines are very important for conducting a research study. They're not always set in stone, and timelines may fluctuate as you are doing your study. But it's good to set out different goals for each of the different activities as you're moving along your research process. And these can always be refined and fine-tuned later on. So one thing that you can do to set up a research timeline is to set up a Gantt chart. This is a bar chart that visibly displays a research timeline, and marks critical calendar dates and deadlines. If you include one in the protocol, it's a good way to promote steady progress towards completion. So figure 16.3 includes a sample Gantt chart for a year-long secondary analysis project. Here you can see it sets up the different tasks in the rows, and then our columns have the different months. So we're saying here that our study starts in July and it ends the following June. So the first thing that needs to be done is actually downloading that public data set and supporting documents, and that is done in July. The next thing that needs to be done is to read the relevant literature. But this is not just something that happens once. So the researcher here has specified that this should be done in July and August, as well as February and March, and the following June. And this is really important to make sure that reading the relevant literature is accounted for throughout the project because of other studies that may be published. And then it goes through and it has all the different study procedures, as well as the final products, writing the manuscripts, presenting at a conference. These are all things that are part of the full research process that should be included in the timelines. Section 16.5 covers roles and responsibilities. Research projects are most successful when all research team members have a shared understanding about each contributor's roles and responsibilities. So it's really important that each of these are lined up, and this is explicitly stated within the protocol. Institutions require one researcher to act as a primary investigator, or principal investigator as it is often called, and to accept responsibility for guaranteeing that the protocol is followed, the budget is managed, and adverse outcomes are being immediately reported, say for an experimental study. Section 16.6 covers preparing for data collection. So once you have set up your timeline, you have your measures set up, you know what you're going to ask in a survey, say, or what data that you're going to be collected from participants or from previous articles, then the logistics should be confirmed before starting your data collection process. You want to come up with this plan for data collection. And some of the things that need to be considered are supplies, equipment, and data collection management systems. This could be software, and it could be purchased, things like Data SPSS, or it could be software that you already have, like Microsoft Excel or PowerPoint or Microsoft Word. So these are all things that should be accounted for and thought about when preparing for data collection. So that's it on research protocols. Thank you for listening.